Well, welcome everyone. I'd like to begin by acknowledging the traditional owners of the land on which we meet today, and I would also like to pay my respects to elders past, present and emerging. Good afternoon everyone and welcome to the October FightMND Scientific Seminar. My name is Sarah Bennett and I'm the Research Coordinator at FightMND. We hold these seminars monthly on the last Tuesday of the month. And last month in September, we heard from Professor James St John, who is the head of the Clem Jones Centre for Neurobiology and Stem Cell Research at Griffith University in Queensland. James is a pioneer for incorporating lived experience into his research and has been awarded by the NHMRC for his community design. I really encourage you to listen to this talk on our website for inspiration on involving lived experience in your research design. So following the talk today, we are leaving plenty of time for discussion and questions. So if you would like to ask anything, please use the reaction button to raise your hand and one of our team will enable your microphone and camera and let you know to ask your question. Alternatively, you can also type your question into the chat where our team will be monitoring and will read out questions on your behalf. Please note that the chat function is only enabled if you are using the app-based version of Teams and unfortunately does not work if you're on the web-based version. Please make sure you introduce yourself and where you're from before asking your question so that we can all get to know each other. We are recording today's presentation, so if you know of anyone not able to join live today, they will be able to watch it back later on our website. So now to the presentation for today. Associate Professor Albert Lee is a group leader of the Neuroproteomics research team at the Macquarie University M&D Research Centre. Albert completed his PhD at the Australian Proteome Analysis Facility at Macquarie University, following which he spent 4 years in the US training at New York University School of Medicine and Johns Hopkins University as an NHLBI Fellow. Returning to Macquarie University, he now leads a team using unbiased proteomics tools to dissect pathways, post-translational modifications, and protein aggregation in MND and other neurodegenerative diseases. And we're fortunate enough to have Albert join us here today to tell us about how unbiased proteomic strategies can provide a powerful tool in MND research. Thank you so much for speaking with us today, Albert, and over to you.
Thanks, Sarah, and thank you to the FightMND team for giving me the opportunity to kind of really share with you some of the proteomics tools that are currently available, and that's something that you may have or have not thought about in applying to your own research. So really, the point of the talk is, a bit more informal and more educational, with less about specific research aspects. But it's really to kind of get people to think about whether proteomics will be useful for their projects, particularly given that grant writing will be coming up towards the end of the year and early next year. It's probably timely to carry out any pilot proteomics experiments that you may be thinking about to either support your current data or something that may be useful later on to kind of generate your next set of hypotheses. And so my talk is about how do we apply proteomics to really map disease networks and mechanisms that may be useful for your research. And so before I get going with the talk, I just want to introduce my team very quickly, who essentially do all the wet lab research and keep the engine room running. I need to acknowledge their efforts throughout the years. And a lot of the members have embarked on this journey with me in establishing and maturing what I now consider the neuroproteomics research program. And so really the outline of the talk today is, why use proteomics? And the outline will be, what is the right approach for my study? Can it help my research or perhaps even my upcoming grant applications? How do we use this information to then validate and generate our new set of hypotheses? And I'll provide 2 case studies that have stemmed from my group. There have been other collaborative projects that have come from my group as well, but the 2 that I will focus on are really using proteomics in an unbiased way to look at pathway changes in disease, but also how we can use proteomics to look at protein-protein interactions to perhaps find new druggable targets. And so in this talk, when I'm referring to proteomics, I'm often referring to mass spectrometry-based proteomics, since there are other platforms now available, such as Olink, SomaScan, which are a bit more targeted aptamer-based approaches. And so, proteomics, can often be viewed as a magic black box that you put in some samples and then output comes out. And hopefully this talk will enlighten you to see whether or not proteomics is fit for purpose for your research. And so really what I'll do here is just run through the basics. So proteomics really is the study of all the proteins expressed in the cell, tissue, or disease state. It's a large-scale characterization of proteins. It is considered unbiased if you look at it from a top-down point of view. And it relies heavily on sequenced genomes and proteomes. And that's the one thing that I kind of want to allude to is that the proteomics data is heavily reliant on having available genomes and proteomes. And so if you're looking at a brand new organism that you found in Antarctica that doesn't have sequenced genome, there are ways to get around how we try and characterize some proteins. Let's see if I can pull up my laser pointer. And so if everyone knows what genomics is, it effectively is study of DNA, and that's kind of fairly static and can tell us what possibly can happen. It is kind of like looking at the blueprints or the architectural drawings of design. You've also got transcriptomics, which is looking at RNA and then tells us what could probably happen. It tends to have a high turnover, and it really, if you want to think of it this way, is looking at the architectural drawings or the blueprint and then really starting to assemble the parts to really kind of get something together. Proteomics tells us what actually happens. It tells us functionally, what these parts, when they're put together, what can they possibly do in the cell or a tissue. And then you've got other aspects as well, such as PTM-omics, which is looking at post-translational modifications. How can these proteins be decorated to perhaps alter their function or alter their solubility? You've also got metabolomics, and that's looking at things such as, what are the metabolites that are changing in a disease, for example, and lipidomics as well. And so when we kind of think of the complexity of the proteome, when you've got the genome, which is approximately 20 to 25,000 genes that have been discovered so far, you've got a number of transcripts that can be derived from that, and we're looking, somewhere in the order of hundreds and thousands, and that's a growing number. But when you look at the proteome, particularly when you're looking at things such as alternative splicing events, truncations, and things like that we often see in M&D biology, and you include the post-translational modifications as well, you often get proteins that that vary quite a bit, and so that number is often quite a large number. And so really the way we kind of look at proteomics is, well, how can we use this information here to really try and capture what is functionally going on with our model system or our disease. And so I'll go through some of the terminology to kind of really provide some context as to what you may come across if you're planning on approaching proteomics for the first time. There is this notion of looking at, top-down versus bottom-up proteomics, which is a term that you may come across. And so if you want to think of it this way, the top-down proteomics approach really analyzes whole proteins directly. It preserves the post-translational modifications. And so if you're interested in a very particular protein and interested in a specific PTM, such as phosphorylation or ubiquitylation, this is the approach that you would take, and it allows you to look at information such as protein isoforms for more of a complete protein structure and function point of view. And so if you want to think of the really old school 2D gels, for those of you who have ever seen one of these, I did a lot of these during my PhD. When we're kind of looking at whole proteins, for example, let's say this was your disease and this is your control. If you see something that is, present here and not here, this is probably something that you would excise to then look at from a whole protein point of view. What are the features of that protein that make it different? The other approach is what's also called the bottom-up proteomics approach. And this involves actually providing a complex mixture usually and digesting up those proteins up into little tiny small peptides and actually putting all that into the mass spectrometer. And that allows for the mass spectrometer to provide broad protein identification, but it loses information about intact protein isoforms. And so depending on the question that you're asking in your project is whether or not you take a top-down approach or bottom-up proteomics approach. We kind of take a more blended approach. We can either use one or the other, or a hybrid of the two, just depending on the type of question that the project tends to ask. The other terminology that you might come across is also something that's called DDA and DIA, or data-dependent acquisition and data-independent acquisition. And these terminologies are based on how the mass spectrometer is acquiring the data. Okay, so data-dependent, so data-independent acquisition. You may have heard the term swath, and that was a term used by one of the mass spectrometry companies from a marketing point of view, but it's effectively more or less similar to what's considered data-independent acquisition. And so what I've kind of put here is just a picture of the top view of Sydney. I had a picture of New York originally, but I don't think everyone's been in New York. So here's a picture of Sydney. And if you want to think of Sydney as, this is coming from the south end, this is the north end, east, and towards the west end. Data-dependent acquisition, what it does is it selects and fragments the most intense ions in real time. And what it does is that it gives high quality spectra, but potentially misses low abundant proteins. So if you want to think of data dependent acquisition, it's kind of like you sitting in a helicopter or you sitting up on the Sydney Tower and actually just looking down. The very first things that you actually start to see are the really tall, huge buildings. And that's effectively what data-dependent acquisition essentially is. It's looking at what are the most common or the most abundant peptides that we can see. And then what it does is that it is trying to accumulate as much of that peptide as possible to try and fragment that into high quality spectra. And so there is a trade-off with the data-dependent acquisition versus data-independent acquisition. So from a top-down point of view, if you're sitting at the top of Sydney Harbour, what you're kind of looking at are the really tall buildings, okay? You can still see that what I've done here is just covered the very small buildings, okay? But if you were to sit at the top of this building here and look down, you can probably still recognize that, hey, this is actually the view of what Sydney looks like, okay? You probably won't be able to see the people walking around, but hey, it kind of gives you a large perspective as to where are the big buildings, where are the small buildings. All the big buildings are on the south end. The North Sydney are mostly houses, and so they can kind of give you that kind of outlook as to what is going on in, I guess, the proteome. Data independent acquisition is slightly different, and hopefully this kind of analogy works a bit better. So using the same kind of analogy of the top-down view of Sydney from the south end, the way data independent acquisition works is that it actually narrows down on what are called little tiny mass windows to then fragment everything within this little tiny window at very small increments. And so what it's doing here is actually fragmenting and trying to isolate everything within this window to try and capture as much information as possible. Now, what that comes up with is a whole huge set of data. And this is kind of the mass spec acquisition method that is kind of a lot more popular nowadays, given that it is able to actually fragment and actually isolate and identify a lot more proteins. So you're able to actually see much more information at the street level in this analogy compared to just looking from the top down. There are pros and cons to both, and I'm not suggesting one is better than the other, but that's just something to be aware of, that is available as two types of acquisition methods. Now, these acquisition methods, when we often approach projects, we often perform them in what's kind of considered a label-free type of way to kind of give us relative quantitation as to, is this building 15 times higher than this building? And so it kind of gives us a kind of perspective or picture as to, hey, this is probably about 15 times taller than this building here, okay? If there is more accurate quantitation that is required, and in some cases it is required, there are other labeling methods such as SILAC, which labels proteins within cells in real time, as well as another approach called TMT labeling. And what that essentially does is that it barcodes peptides so that when you kind of mix the samples together, they can be run once in the mass spec to reduce variability. But that kind of gives you much more accurate quantitation, given that what you're quantifying are these little tiny barcodes that are then attached onto these peptides specific to the samples that you're being analyzed. The caveat obviously is that there is a much higher cost for the reagents and labor time, but there are also other methods such as absolute quantitation if you wanted to spike in a peptide. And so really, when it comes to proteomics, data quality is really highly dependent on good sample preparation. And so a typical workflow that you would go with is, if you've ever worked with proteins in your samples, for example, like running a Western blot, you've already done half the sample prep, okay? So often than not, when we kind of get collaborator samples in, the samples are already made up in, Ripper buffer, okay, because it was used for a specific type of analysis, such as Western blots and so forth. We can repurpose some of that to actually perform proteomics experiments. And so if you've got, in this case, let's say sample 1 versus sample 2, you can run that in a gel like you would for a Western blot. We can then purify the proteins and perform trypsin digestion to then analyze that on the mass spec, and effectively it gives us this kind of information. Now that information can then be analyzed using software, either commercially or open source software. But effectively what that software is doing here is trying to match to see what these little tiny sticks are, and what are the specific amino acids that they belong to. Now, it's not too different as to those who have ever played the game of Sudoku. It is really trying to then use a software tool to try and analyze and match what is going on, to determine what the peptide was that was being analyzed, and the intensity of this peptide, for example, is a relative measure of the abundance. So the higher the intensity, generally speaking, we say that, hey, actually, there's a lot more of that in your sample. And so when the software then does its computational analyses, it can then kind of give you information as to, differential expressed proteins, which you can then, start to generate heat maps in code, or there are freely available web tools to allow you to do that. And then using that information, you can then start to map out, okay, well, all these proteins belong to X pathway, okay? And so that kind of gives you information as to, okay, well, if you have a question that you wanted to try and perform unbiased proteomics, it kind of gives you a lead as to where to chase. And what will happen with a lot of these leads is that, yes, there will be multiple pathways that are changing, or multiple biological processes that are changing. But because we're M&D biologists as well, we're able to kind of filter out what are the important ones and which ones aren't, to kind of give you, I guess, a leeway as to, this is the pathway that we should chase. And so really, to kind of finish up the first third of my talk, and I'll go through this really quickly, there are some things to consider: DIA or DEA or both. You can actually do both, not together, but you can do them one after the other. So the The pros and cons to both approaches. So DDA, or data-dependent acquisition, captures most intense ions. It's great for discovery, but biased towards abundant proteins. And it's that notion of you sitting on top of a helicopter and looking at the top down of a CBD and really just looking at the bigger buildings. It does give you still that information as to where you are, but it doesn't capture everything that's down in the grass. There's also data-independent acquisition, which acquires all ions within set mass ranges. It improves reproducibility and quantitation, but requires more data processing. So there are pros and cons to both. As I mentioned very early on, it relies on search algorithms, which look at which are just software packages that look at the mass spectra to really try and make interpretations as to how to translate that spectra into peptide information, as well as sequence databases. And in most cases in M&D research, because we're working with humans, sequence databases are quite thorough, and so that's something that isn't usually an issue. There are several assumptions and parameters are made when searching a lot of the data. As I mentioned, trypsin is the most commonly used sequencing enzyme that are used to make peptides. Now, if you're looking at a protein, and I use the example of TDP43, if you're interested in, for example, the C-terminus of TDP43, which doesn't have many arginines and lysines, trypsin may not be the right enzyme to use. Okay, and so we may need to actually look at what are the other suite of sequencing enzymes that may be able to capture the C-terminus of TDP43. This is also the case with some cryptic peptides as well, where some cryptic peptides may not have arginines and lysines, and so therefore you may need to look at, what are other enzymes that may need to be used. Chemical treatments as well. If you're using, for example, loads of urea and heating it, there are certain things that may modify amino acids that we may need to be taken into consideration, particularly when searching the data, given that that may shift the masses of amino acids. Sample preparation is key to obtaining good data, and so polymer and keratin minimization, lysis buffers. So all our lysis buffers that we use have some sort of detergent, and they're considered polymers, which can really contaminate or even overcome the peptides that are in the mass spectrometer. It generates huge amounts of data and requires statistics to generate multiple leads. While having a lot of data is great, there is a lot of caution to that. Having too much data can be detrimental as well. And so there requires a lot of thought in the study design at the very first instance before going ahead and actually generating a load of samples. Okay, you kind of want to make the design of the study quite thorough, and with the right N values, with the right combinations at the very first instance before going ahead and growing things up. Dynamic range of proteins in complex samples can cause a mass spectrometer to miss proteins, and I've used the example of plasma here, which is notorious for that. Reproducibility for both biological and technical. Often with any omics technology, we often want to run samples all in the one go. So if you have your n equals to 3 or n equals to 6, you kind of want your samples prepared in such a way that all those samples are ready to go, prepared and analyzed from day one. You don't want to be analyzing n equals to 2 this week, n equals to the next n equals to 2 3 months later, and n equals to 4 6 months from there, only because you will get variability in the results. And so what I kind of want to illustrate here is there are some applications that we commonly use in M&D research, and it's probably some that will be handy or you may find useful for your own research. And these are the kind of the main approaches that we've used in my group here. And that's really looking at global proteomics, and a question may be, hey, what is the difference between sample A versus sample B? protein interactomics, what are the proteins interacting with my protein of interest, and what are the friends and clusters of friends that are hanging around my, my protein of interest. There's also post-translational modifications, and you may be interested in how do PTMs affect the function of your protein of interest. Is a specific pathway activated by a cascade of phosphorylation as a consequence of kinases and phosphatases? There may be questions that you're asking. The other ones, plasma proteomics, tissue proteome profiling, which I won't go into because they kind of need a bit more. They're workflows that we work with, but they are all collaborator and project specific because there may be a very specific protein in a piece of tissue that a researcher may be interested in. So certain layers of optimization and study design are required for that. At the moment, my lab houses 2 mass spectrometers, an Ultimate 3000 nano LCQ Exactly Plus, as well. We recently acquired an Explorers 480. And this is run out of our team at Macquarie, and we only focus on MND and neurodegenerative research. And so these are mass spectrometers that are dedicated just for MND research. So these are really our workhorses. These are our Toyota Corollas that are reliable, our workhorses, and it just gets us from A to B to give us an answer. And so I'll just go into the MND side of things. And so the 2 examples I will kind of talk about are really TDP43 and this other gene called CCNF. And so to kind of switch gears into how do we apply proteomics for ALS research. And so everyone is very aware that there is convergence of genetics, mechanisms and pathways between ALS and frontotemporal dementia. We now know that the pathological feature of ALS is cytoplasmic TDP43 aggregates in cells or in neurons. And this can be due to a number of different pathways that are affected. There are numerous papers that have shown mitochondrial dysfunction, autophagy, impaired axonal transport. All these pathways are somehow involved in the outcome of neurodegeneration. However, the challenges with research, and everyone more or less will know this, is that the models that we kind of work on are difficult, particularly with neurons. They're post-mitotic cells. Diagnosis of MND is not confirmed until post-mortem, and the current models capture some features of disease. And so when we kind of view this in a very simplistic model, when we approach this, we often lead with the notion that perturbations and proteostasis dysfunction contribute to pathological features that lead to cell stress and eventually death. We often start with a candidate gene mutation, stress, treatment, or model that you're interested in and determine what type of stressful survival is occurring. This can either lead to, proteostasis dysfunction or restoration, depending on the question that you're asking, and if TDP43 aggregation or stress granule formation or restoration of these aggregates is also a readout that you're interested in. But eventually, the models that we often use lead to some sort of neurodegeneration or apoptosis. So really, in an attempt to kind of tease this apart, we usually use the approach in a very unbiased way to kind of use global proteomics to really identify what are the differentially expressed proteins, which are the buildings that are the big ones and which ones are the small ones. And to try and use that information to determine where are the traffic jams that are occurring in the CBD. And that kind of gives us as the lead as to, okay, well, this is where to look. There will always be a number of cell pathway changes, and it's usually up to the biologist to really determine which cell pathway is important and relevant to the question. For example, if you're planning on examining a drug that targets, the mitochondria, for example, but the model that you've employed shows perturbations to nuclear import and transport, that may not be the right model to use for, a drug testing experiment. And so this type of information kind of gives, information on the mechanisms, and potentially other avenues to validate or restore the mechanism. So if a cell pathway was shown to be perturbed, if you administer a theoretical drug that you think may restore, we can use proteomics to see whether or not that that pathway has actually switched or turned off or turned on, what have you. And lastly, I guess for scientific rigor, it's also ideal to examine that biology in other appropriate models as well. And so the example I'll use here is one where we've used global proteomics, and this is a very a study that looked at CCNF mutations in the family with ALS and FTD. So Associate Professor Kelly Williams and Professor Ian Blair discovered this CCNF gene mutation in a multi-generational family that passed down this S621G mutation. And so when I was first recruited to the M&D Center, I was actually asked to determine what this mutation does. Okay, what is this one mutation? What is it doing from a functional perspective? And long story short, it turned out that the original S621G mutation was a phosphorylation site that switches off the enzyme. So this enzyme here, this CCNF, encodes this E3 ligase enzyme, which ubiquitolates proteins. This phosphosite here was one that switches off the E3 ligase activity, and the fact that because it was mutated in this family, this phosphorylation site conferred an elevated activity, ubiquitylation activity, which eventually caused defective autophagy and a buildup of ubiquitolated proteins. And so that's really old work. But really, the next question was to ask, okay, well, what are these other mutations actually doing from a functional point of view? They're found in ALS patients, but what do they actually do? Some in blue were found in databases, and it was unclear whether or not these were actually ALS causative. And so the way we kind of did this was using a global expressional proteomics profiling of these mutations. And so all we did with this was made all these mutations in little tiny constructs, put them into cells, so transiently transfect these into cells as a very simple plug and play approach, if you want to think of it that way. And these were just inserted into PM cherry constructs. And then we carried out global proteomics. So we just said, hey, we're going to transiently transfect it, lyse the cells, extract the proteins, and then run it on the mass spec. What we found then was that there was a number of different pathways that were changing commonly and uniquely between the different mutations. And this kind of gave us scope as to, okay, well, what are the different pathways that are changing with all these mutations and how are they affecting it? Now, that's looking at from a top-down point of view and just telling us, okay, well, these are all the bottlenecks or all the traffic jams that are occurring in the cell. And so what we found was that these three mutations, K97R, S195R, and S621G, were all triggering apoptosis signaling. So that kind of gave us a clue as to, okay, well, these three mutations are highly important because they're actually causing or leading to a potential apoptosis phenotype. There are other pathways as well that are kind of involved in ALS. We didn't chase them all, but this kind of gives you information as to, okay, well, perhaps I'm interested in active cytoskeleton signaling, and I am looking for a model that shows elevated levels of active cytoskeleton signaling. Perhaps I would choose this model to then, test my hypotheses. And that kind of gives you very powerful information to really look at the study design from that point of view as well. And so this is the kind of information that comes out of proteomics data. It's not too dissimilar to transcriptomics data, where you've got a gene name, fold change, and a p-value, and you can kind of then start to map out, well, what are the pathways that are changing? And so in this experiment here, I mentioned K97R, S195R, S509, S621G. What you can see here is it's bright red. All these proteins are elevated, and we can kind of map that to the apoptosis pathway as an example. Now, this is all omics data. It's always good to then try and validate that either functionally or using much easier assays. In this case, we use Western blots to just prove that, hey, actually the proteomics data, it's pointing to the apoptosis pathway as being defective. And so therefore we validated the intrinsic pathway of apoptosis using phospho-Bad amongst others to validate the proteomics data and the predictions as well. What I haven't shown here as well is that in addition to the apoptosis pathway, the ubiquilation pathway was also different amongst the different mutations, and you can perform functional assays in this context as well if there is available, if a company sells a kit that says, hey, we can measure the activity of X protein, that is also your next validation step that you can also take as well. And as I mentioned, you then validate that in other models. So this is work from Dr. Claire Stevens and Professor Lizanne Uy, where they had human-derived iPSCs from S61G patients, and we also showed similar pathway changes in that. From a phenotypic point of view, work from Dr. Alison Hogan also showed that these mutations caused apoptosis of spinal motor neurons and cause motor deficits. And I think at the moment there's work being followed up in mouse models with Professor Lizanne Uy as well to see this phenotype as well. So does this approach work? We've also applied this to global proteomics of TDP43, A315T, and M237V, where you can just plug and play to kind of give you really quick information as to, well, are these pathways changing? And from this information here, long story short, is that this showed that these two mutations cause perturbed DNA damage responses, and there've been numerous papers on TDP43 mutations causing impaired DNA damage response. So I'll try and switch gears and try and go as quickly as possible, and to kind of switch gears into what other questions that you can ask. Protein interactomics is a huge one that is now being explored quite a bit. And so we've kind of used this proteomics approach to try and look at who are the friends that are binding to TDP43. So this project here was to identify what are the TDP43 protein-protein interactions in the nuclear and cytoplasmic compartments, as often one of the features of TDP43 is the mislocalization of this protein. So we hypothesized that perhaps a transporter might be involved, and we wanted to carry this out in a very unbiased way. And so there are numerous papers that show that there are defects to nuclear cytoplasmic transport of TDP43. And so the way we kind of approached this was we used an APEX2 proteomics workflow in both Neuro2A and primary neurons to map TDP43 interactions. And so the way this approach works is that we encode an ascorbate peroxidase with TDP43, which then sprays a bunch of biotin around all the proteins that are interacting with TDP43, and then put that onto the mass spec to try and identify proteins. We can also perform antibody-based pull downs and put that on the mass spec to identify what are the co-interacting proteins as well. And so this is work coming out of Professor Adam Walker's group, where we find that with stress as well, the interactome of TDP43 in the nucleus and the cytoplasm are completely different. And they are actually quite different when you compare it to the whole cell lysate interactome as well. And this on the left here is just illustrating that when you oxidatively stress cells, you get a number of pathway changes that occur, mitochondrial dysfunction comes up, but also RNA metabolism also appears there as well. And from the Western blots that you can kind of see on the right-hand side here, you can kind of see that the different proteins that interact with TDP43 are quite vastly different between the cytoplasm and the nuclear fractions, and they actually change with oxidative stress as well. And so this is really to kind of illustrate that, hey, if you look at compartmental interactomes, they will be different to when you perform a pull down of a whole cell lysate. And this is what this is illustrating here. You can then do gene ontology analysis to see, okay, well, are the proteins in the cytoplasm, are they mitochondrial proteins that are interacting more with TDP, or are they lysosomal proteins more? Okay, so that kind of gives you that kind of depth of information. As I mentioned, you can perform simple analyses and validation steps with IPs and Western blots. And the take-home message from this was, from this paper from Flora, was that a number of different proteins interact differently in the nuclear and cytoplasmic compartments with mutations, as well as with oxidative stress. And so some of these targets are perhaps druggable or interacting targets that may be interesting and may be looked at for future work. And so to really summarize my work here and why you may think about using proteomics is that it is powerful and versatile to answer and guide diverse biological questions. It's always good to come up with a clear hypothesis and use the right models and design appropriately. And you can kind of sit down with a proteomics scientist like myself to kind of come up with a study design that is appropriate and fit for purpose that answers your biological question. Validate findings with orthogonal methods. Proteomics has caveats. It measures relatively fold change of protein expression. It doesn't look at things such as activity or functional activity. So if you're interested in an enzyme and you want to know, hey, is this dysregulated? The abundance of the enzyme might be the same. However, functionally, from an enzyme point of view, it may be completely different. Now, we do have tools to kind of use that information to infer whether or not the enzyme activity has changed, but there's nothing direct that will measure that using a proteomics approach or from the very first instance. So in neurodegenerative research, such as MND, proteinopathy and proteostasis dysfunction, dominate, and proteomics can help in mapping disrupted cellular pathways under stress. You can quantify interacting aggregating proteins. You can also characterize PTMs if that is of interest to your disease or protein of interest. And with that, I just want to quickly thank the funding support, FightMND, for giving me this opportunity to kind of provide an educational talk to those who may be interested in wanting to employ proteomics for their research. And with that, I will take questions. Sorry, I went over time, Sarah.
Oh, not at all. Thank you so much. That was so informative and really inspiring. I hope hope you've inspired some people to really look into this approach. So we're opening the floor for questions now. So just to the raise hand button at the top of your Teams app to raise your hand, and I'll enable your microphone and camera for you to ask a question, or else you're very welcome to type your question into the chat and we can read it out on your behalf. So I guess while we're waiting for some questions to come through, Albert, I'll just ask if I've been inspired by your talk today, but I know absolutely nothing about proteomics or where to start, and my supervisor's not very experienced in it either. Like, what's the first step?
I think the first step would be to have a casual chat with a proteomics researcher in first instance. You'll be surprised based on the question that you may be asking. Proteomics may be able to help give you some guidance in the very first instance. It probably can't answer the question straight away, okay? But it actually can, I guess, narrow down where to then take it. And yeah, that would be the very first conversation to have with the supervisor is, okay, well, often with projects, it's often where do I start? And proteomics can be a very good starting point.
Yeah. Okay, thanks so much. Gethin?
Hey, Albert.
Hey, Gethin.
A couple of questions. What sort of sample numbers do you need to get any usable data out?
Okay.
Is that how long is a piece of string question? I don't know.
No. It's a great question, and it depends how good your sample is. But if you can run a Western blot, that's more than enough for a mass spectrometer. Mass spectrometers can go down to the nanogram, picogram level of protein amounts. Now, if you're giving someone picogram amounts, but it's also filled with other stuff, that's where it becomes a bit more complex. But generally speaking, when we kind of request samples from collaborators, we often say, if you can spare us, 10 to 20 micrograms more, that'll be great. That's more than enough for us to run analyses. And that's usually like one Western blot. And often the case is we will have usually collaborator researchers who will, have samples ready to run the Western blot, but then may spare a little of that same sample to run proteomics. So you're kind of getting two experiments out of the one tube, if I'm making sense.
What about numbers of replicates as well?
That also depends. If it's an initial screening, like a good starting point, like what Sarah was asking, and n equals to 3 between the two to get us going would probably be the good starting point. That's with cells. With animals, for example, I would say an n equals to 5, 6 as a minimum starting point. The reason being is because we have seen a lot of variability just between individual, mouse or animal samples. Because you've also got sex differences as well. And that actually makes a huge difference in proteomics spaces.
And if you were, it's a really naive question, since you're going to be chopping up the samples anyway, as in tryptin or whatever degradation, does it matter if the samples are a bit degraded already, if they've had a tough life, been freeze-thawed a couple of times, or been stored in an unreliable freezer somewhere?
We prefer, that makes it more challenging to work with. But then with degraded samples, I don't think much can be achieved, proteomics or not, with those type of samples. You would still be able to get some data. However, there will be a lot of truncated proteins, chopped up proteins that may not be able to achieve the full protein coverage or proteome coverage. So what I mean by that is typically from a cell sample, you might get 5,000 protein identifications. From a degraded sample, it may be half of that. So sample quality does matter. And I think that's also the case with any other biochemical technique as well.
But if you can't get full coverage, is it still better than nothing sometimes? If you get partial?
It will still give you information. Now, whether or not that's the information that you're chasing is another thing. Because often or not, when we get people with their projects, they often have, they often will already have one or two pathways to go with.
Cool. Thank you.
Thanks, Catherine. I'll just throw to Double for some questions.
Thanks, Sarah. Thanks, Albert, for the presentation. I'll admit I'm coming in very naive, so I apologise if some of these are a bit too simple. In terms of data, you had one of your slides saying, often there's the danger of, coming into an experiment and then generating, a lot of data. And my question is, some of it's not specific to your question. Like my question is, like, what ends up happening with that excess data? Does it ever get utilized ever again, or is there a way to then redefine your questions and come back to the data that already exists?
So we've had numerous circumstances where we've analyzed like a bunch of data and then had to split that up into two separate studies. That's a very common occurrence because the too much data probably is too much for one specific story or one specific paper and needs to be split up. And so it does get repurposed for the exact reasons that you're suggesting, where you've got so much data, but perhaps it doesn't all fit into the one hypothesis or the one story. And so it can kind of be split into two, and then the data that's not used can funnel the next set of projects.
Mm-hmm.
Great.
Yeah. Can I just one more question, Sarah, if there's, I'll try to make this succinct and clear. One of your slides, you were talking about the potential for biomarkers and identification, following disease progression, perhaps. I just coming back to Gethin's questions about how much sample is actually needed to be able to verify a biomarker or even target engagement, for example, of in a clinical study, for example, in terms of clinical setting, in terms of being able to identify target engagement, but also then how quick does this analysis occur? Is it appropriate for a clinical setting to kind of assess, or a precision medicine approach to assess whether a treatment is right for a particular kind of individual or not? Are there options there?
I mean, that's a tough question, and it's an appropriate question. I mean, the mass spectrometer is only as good as what it can detect. Now, if the biomarker of interest, then we're talking about a hypothetical biomarker. If it's down in the grass in a biofluid such as plasma, it may be difficult to utilize mass spectrometer for that purpose. If it's if it's something that is readily seen in a biofluid such as plasma, mass spec can be utilized for that. Now, from that point of view, particularly for the clinical space of things, the mass spectrometer will probably need to be NADA accredited so that it is appropriate for clinical use with absolute quantitation. At the moment, the mass spectrometers that we've been using have been used for, I guess, discovery type work, predominantly. For clinical work, it is still possible. However, the biomarker the hypothetical biomarker would need to be carried out in a more targeted way. And we can kind of do that with the mass spectrometer. So with a mass spectrometer, what you can do is, let's say you've got hypothetical protein or hypothetical biomarker that has a mass of 7,000 Daltons. You can kind of put that and type that into this mass spectrometer to kind of look for that. So what I mean by that is you filter out everything else. It's kind of what's called a PRM approach, or a parallel reaction monitoring approach, where you kind of remove all the background, and I'm only looking for my hypothetical protein at 7,001 Dalton. And so it is possible to do that. It just requires a bit of optimizing and knowing what target to look for.
Thanks, Albert.
Thanks, Albert. I just have a question in the chat from Deepika Dixit. It says, hi, I'm a PhD student at the Wicking Dementia Centre at the University of Tasmania. I'm currently exploring options for proteomics analysis software, and I'm wondering how to choose the most appropriate tool for my research. I've used Perseus and GSEA, but I'm not sure how to best decide which software best fits my needs. Do you have any advice?
Yeah, I mean, I can probably take that offline. Because there is a lot of different open source software and the commercial software as well. Mm-hmm. It also depends on whether or not the data they've acquired is data dependent or data independent.
Yeah.
As well. So I can probably take that offline if they shoot me an email.
Yeah, so Deepika, if you wouldn't mind shooting Albert an email after the conclusion of this seminar, I'm sure he'll be more than happy to help you out. Please reach out if you need his contact details or anything. Okay, I might just wrap it up there because we're just going to throw over to Davo again. He's just got a few words about the Longitude Prize on ALS.
Thanks, Sarah. Yeah, as Sarah said, I'll share a few words about the Longitude Prize on ALS. Just reminding everyone that stage 1 applications are closing on the 3rd of December 2025, and starting off by saying the Longitude Prize on ALS is principally funded by the MND Association in the UK and designed and delivered by ChallengeWorks, and is also supported by FightMND and the other organisations listed below. The prize is a $7.5 million international program that is incentivising AI-based approaches to transform drug discovery. The prize aims to identify and validate new therapeutic drug targets, and provide participants access to the largest data sets in ALS and providing computing power and data support. The data sets that are being provided currently include Project Mine, Answer ALS, ALS Compute, the data from the New York Genome Center and ALS Therapy Development Institute. And data support is being provided from the AWS and DNA Nexus. And the prize is seeking collaborations between AI biotech, computational biologists, and researchers to form teams and submit applications. As probably most of us know, the prize has launched, and applications for the first stage of the prize, called Discover, which is focused on using AI to identify new high potential targets for ALS, are due on December 3. There are 2 further stages, with the winning team then ultimately receiving £1 million for finding the target with the strongest evidence of therapeutic potential. More details on the Longitude Prize website or through the QR code on the right. But really important to register for the Longitude Prize and contact Challenge Works, especially with regards to collaborations. They will help you find potential collaborators to form teams, whether that's in the AI space, biotech, or research teams even, and to enable you to form strong applications. With that, and to help with preparing your applications, FightMND would like to invite you to a webinar on Thursday, the 6th of November at 5 p.m. Australian Eastern Daylight Time. The webinar called AI Meets ALS. And, the webinar will highlight the importance of the APAC region and participation of the APAC region in the prize to kind of ensure expertise in our part of the world are recognised on a global stage. It's also going to highlight the transformative potential of AI in accelerating MND drug target discovery and the importance of cross-sector collaboration. There will also be advice and tips on preparing your application, and this is from a prize judge perspective. We'll have prize judges involved and presenting, and these include Professor Amar Al-Shalabi from King's College in London, Dr. Nortina Shefrazela from the University of Malaya, and also President of Pactals, and Dr. Sophie Nyberg from the MND Association in the UK. So please register for the November 6 webinar using the QR code there, and be ready to ask questions to the presenters, to the prize judges, to help craft and strengthen your applications. Thanks, Sarah.
Thanks, Davor. Please reach out to him if you do have any questions about the Longitude Prize or these webinars. And thank you again, Albert, for joining us today. I thought that was really informative and gave a lot of people good food for thought in terms of their upcoming applications and how proteomics might help strengthen and give new hypotheses for those. If you are interested in incorporating any of these unbiased proteomics techniques into your research program, I'm sure Albert would be really happy to have a chat to you about how he can help out. So thanks again for joining us today for the September FightMND Scientific Seminar. We hope you've really enjoyed it. Shortly after we close, I will actually be sending out a brief survey to everyone who's registered for a seminar this year, just to help us understand what you get out of this series and what you'd like to see moving forward. The final seminar for 2025 will be on Tuesday, the 25th of November at 12 p.m. Australian Eastern Daylight Time, and we'll release the details in the next couple of weeks, so keep an eye out in your inbox for this. Until then, all the best, and we'll look forward to seeing you next time.